Hi everyone, today I'm going to demonstrate our ChemScraper tool which is used for parsing molecular diagrams. So first let me give a quick overview of the tool. So the main task of the ChemScraper tool is to basically parse molecular diagrams from documents. So the input is a scientific paper in the form of PDF, which contains molecular diagrams in basically two forms, which is embedded raster images, which can be JPEG, PNGs, or the other image formats embedded in a document, or it can be drawn vector instructions, which contain all the vector information available in the PDF itself. The output we produce is basically CDXML, which is a XML format for representing the chemical graph um, for each molecule diagram that we parse. So this represents both the visual as well as the syntax, the chemical syntax of the molecule diagrams. And the CDXML can be converted to smiles, or it can be rendered as SVGs or imported to a ChemDraw file directly. Some of the highlights of our features include the ability to view each extracted molecule diagram with its parse structure in SVG and SMILES format alongside the original context on the document page. Additionally, users can export CDXML for all molecules or a single page or the entire document, which can be opened directly in ChemDraw for seamless integration with molecular structure visualization as well as editing. Users can also export CSV files for the parse results containing detailed metadata about the extracted results. Let me demonstrate this tool. As you can see, you have two options. One is to upload the PDF, another is to use example PDF. So when you upload a PDF, you have the you can provide an email for getting notification on when the results will be ready. You press the Analyze button, the ChemScraper job begins. Depending on the number of pages in the PDF, the time taken varies, but you can save the job ID or the link here and come back to this page later on to view the results. If you provide an email, you also get email notification on when the job is ready. When the results is ready. Um, so for the time being, let's use an example PDF. Press the Analyze button. You can see the results are available. Um, so the results are in the form table where you can see individual extracted molecules from the documents. So the molecule name, if available, the extracted structure in the form of SVG, the original structure that is cropped, that is extracted from the PDF. Uh, these are directly from the PDF itself, and the predicted smile string and the flag column that is used to flag molecules of interest to you. By default, this is sorted in the by the location in the PDF in the top, down, left, right order, but you can change that if you want to sort by molecular weight, the number of atoms, the number of occurrences of each molecule, both in ascending and descending order, as shown here. You can also limit the results based on what is interesting to you. For example, you can use molecules with BR C, uh, and you can see the results being shown which are relevant to this. You can do this using the diagram as well. You can edit this diagram. You can use the periodic table here as shown, or you can use the different tools here to construct the smile string that you want to search, and you get the results based on that. Now, let's look at each individual results. So for example, let's expand this by clicking on this. So you can see that there are additional information for each of the extracted molecule. We have the name, the location in the PDF, the other occurrences of the same molecule in the document, other part of the document. Uh, so these are the information like the molecular formula, molecular weight, chemical safety are pulled from database called PubChem. So PubChem is basically a free open source open chemistry database from the National Institute of Health that provides comprehensive information on chemical substances and their properties, which are sourced from diverse contributors worldwide. So it's a huge collection of molecular information. So using the predictive smile strings from our system, we use PubChem database to pull this information like the molecular formula, weight, chemical safety if available. Uh, One of the more interesting features is that we can see the original molecule or structure in the PDF in context highlighted here in purple as shown. You can open this in the PDF viewer and see all the molecules that were extracted from the original PDF, as well as we can see the current one that we are viewing currently as shown. you also have an option to export to CDXML or CSV directly. So in CDXML, you can export by individual page or the entire document. For CSV as well, you can extract the entire set of molecules, or you can use the current table view if you want to look at a specific view of the table where you are to limit the results to a certain number of molecules based on your search criteria or filter that I'll talk about. So once you export the CDXML, you can open the CDXML in ChemScraper as shown here. So this is the input PDF that we processed, and on the right and on the left we have the CDXML that we predicted by the ChemScraper opened in ChemScraper directly. So on ChemScraper you can view these molecules directly, which are corresponding to the input PDF, and you can edit them directly as well in the ChemScraper software, where you can use for other purposes or extract or export to different formats as needed. So you have all the pages available as shown here for each page of the PDF, which corresponds directly to the individual molecules in the input PDF. So this helps uh, to basically provide operate on these molecules directly based on the input PDFs. You can also export the CSVs, where CSVs provides all the metadata related to the predicted molecules, like uh, the page numbers, the file path, the document, the atom count, the name of the molecule if available, the smile string, the SVGs as well, the location in the PDF, with using the bounding boxes, the PubChem ID that you can look up, the molecular formula, weight, chemical safety, the description if it's available, the page number as shown, the other instances if it's available in other part of the document, and the molecular fingerprint that is used for similarity comparisons and other purposes for each of the molecules that were extracted. Now let's look at the flagging feature. So if you are interested in some of the molecules that are of more interest to you, you can flag them using this flag button. And then you can use this filter option where you can filter by if the molecule name is available or not. If the molecule has a converted CDXML structure or not. In this case, all the molecule have the corresponding CDXML. And if it's flagged or not. So if you want to just view the flagged molecules, you can enable that. And based on this, you can also export only the current table view, that is only the molecules of interest to you, and export that and view that later as shown as we demonstrated earlier in ChemScraper or or CSV. So the code base is available open source. It's open source, and both the front end and back end code base is available. If you click the links here, this is the front end code base with instructions, and the back end code base with instructions to install, um, uh, have make examples to run the pipeline on available in example PDFs or on your own PDFs. So all the information available. All those you can also run it through Fast API on your local server or deploy it based on your needs. As all the examples are provided. The citation information is available for the paper as well. If you click the link, you can also view the archived version of the paper, which is available here, as well as the published version of the paper in Springer, which was published in the International Journal on Document Analysis and Recognition in 2024 as a special edition of the conference. So yeah, that's all. Thank you.